All right, good morning, everyone. Just to clarify, the session is the title, The Evolution of Cores. But before we start the session, we have our sponsor presentation for a few minutes, and after that, we will start. Thank you.
Thank you. So thanks for having us here today. My name is Manisha Ray. I am on the scientific affairs team at DeepCell. And today I'm going to tell you a bit about how we are trying to make morphology the next way that we can analyze and characterize cells to unlock new insights into their biology. So it's well known that cells are, their morphology is inherently tied to their function. So the shape of the cell often determines what the cell will do. So an example I have here is stem cells being differentiated into neurons. When they're in their stem cell state, they physically look round, they grow in these colonies, and that reflects their pluripotent nature, that their function is to be fairly undifferentiated. They don't have any particularly exciting morphology. Whereas their final form of differentiating into neurons, they have all sorts of morphology. They have axons, dendrites, interesting shapes, and that inherently is connected to their function of interconnecting and communicating between cells. And neural progenitor cells, which are kind of intermediate, have a lot of intermediate morphologies and as well as markers and functions between stem cells and neurons. So it's not a shock that morphology is very important to the function of a cell. However, the technology hasn't been there to actually quantitatively study morphology. Most morphology is described in very qualitative terms, in terms of shape, and very descriptive language. So what DeepCell is doing is turning cell morphology into another quantitative metric of biology, similar to the way we look at genomics, proteomics, and other genomic methodologies. So what we're trying to do is add morphology into that high parameter, high content, highly quantitatively analyzable type of modality that we can analyze alongside these other omic technologies to understand new insights about cells. So our platform is an all-in-one system, and the real power of our platform is in these AI models. So our benchtop instrument can take individual pictures of cells, which are then analyzed by these AI models to look at many aspects of morphology, where they can then be sorted into up to 6 different wells. And we have a software suite all connected to the same system that enables you to understand the high-dimensional morphology data that our system produces. What this actually looks like in terms of hands-on is that cells are dissociated into a single cell suspension, so how you would for any other single cell type technology, and then injected into our chip. While in the chip, the cells are imaged in flow, and those images in bright field are analyzed in real time for dozens of different aspects of morphology. And we look at this in 2 different ways. We look at the what we call the morphometric or computer vision-based features, which is what you would typically think of when you think of morphology, shape or area, radius, those types of things. But we also look at dozens of different AI and deep learning parameters, which are not as interpretable, but allow you to get much more granular and quantitative insights into the morphology of cells. And all of this happens in real time, so the algorithms decide the characteristics of the cell based on the image alone, completely label-free, and then it can sort them based on only what the cell looks like. So this can work with viable cells, it can work with fixed cells, and that means once it's sorted, you can use it for any other downstream application. You also get all of this morphology data to play with and look at subpopulations and clustering, and you can look at it in the ways that you would typically look at any high-dimensional data set. This technology can be applied to get greater insights into many areas of biology, and they are very heavily overlapping with the areas you would typically see in, say, single-cell genomics. You can get sample-level information, such as characterizing the profile of the types of cells present in a sample. Very common application of single-cell biology, or single-cell genomics, but here we can do it just with the image. We can also use morphology to detect and monitor diseases and monitor changes in morphology that have been induced by drug screening, CRISPR screening, other types of perturbations. We can also get individual cell-level information, such as cell health. You can use morphology to get much more information on how happy the cells are than you could typically get from something like, say, trypan staining. All of this, the real power, I think, is integrating this information with all the existing data that exists in your cell type, such as genomics, proteomics, and other data. And really, the real power comes from being able to use this label-free method to use morphology the same way you would look at phenotype through any other system, but in this non-destructive, completely visual-based method. So I'll go through a few examples now of how our technology can be applied to different research questions. The first one I'll go through is using morphology to characterize the cell types present in a tumor type sample. So here I'm showing a mix of tumor cells and immune cells, including cell lines and melanoma biopsies. So these are a mix of individual cell lines and actual tumors. And what you're looking at here is a UMAP projection. So all of those dozens of different morphology features can be plotted in a UMAP, which is typically used in things like single-cell gene expression. So every dot in this UMAP is the morphology of a single cell. So this is all morphology, what you're looking at here, not genomics or other data. And here you can see that you can cluster it in the same way you would do with genomics data. And the cells do indeed cluster by cell type. So the melanoma cells are in green over here. The stromal cells are in brown over here. And on the right side, we get a whole bunch of different immune cells. And we can do something called lighting clustering, which is an algorithm-based way of clustering the cells, and overlay them with the actual images, which is a very cool aspect of the system. For every individual dot you see on here, you can connect it to the image that came from it. And you can see here the diversity of morphologies present in this mix of immune and tumor cell samples. And again, we see that the right side of this UMAP has most of the immune cells, but we can find differences in their morphology using this combination of AI and morphometric-based group of parameters. And then an interesting thing to note is that the melanoma cells actually clustered into two separate clusters, and the main differentiator between them is that the upper cluster of cells are pigmented. And this is another example of something that you can see very easily by eye. Melanoma researchers have long known that some cells are pigmented and some are not, but it's not something that's been easy to study because there's not good markers to separate those cells out of the rest of the population. Something that can be done very easily with a morphology-based separation. Another example I'll show comes from a paper we recently published in Modern Pathology. This is our first paper. And here we use morphology to profile the cells present in malignant fluids from cancer patients. So there's an unfortunate side effect where in cancer, you get this buildup of fluids, and in that fluid there can be cancer cells in there, and that can be indicative of prognosis. So we were able to take those fluids and sort out the tumor cells for further analysis, and that's what you're seeing here. So this is a UMAP of different tumor cells that have been pulled out from the malignant fluids of two different patients. And the two different, within each patient, the cancer and the non-cancer cells cluster separately, as you would expect based on morphology alone. And the interpatient heterogeneity is clearly visible as well. The two patient cancer cells are not clustering together. And again, you can see the morphology heterogeneity. And to demonstrate the enrichment of these cancer cells, here we're looking at a CNV profile. So once the cells were sorted on our system, we did genomics analysis, and we see that the genomes of the cells sorted by our system are heavily disrupted. There's a lot of alterations here, indicating that we enriched for cancer cells. And you can also see that here is a list of different samples and the allele frequency of the cancer cells, or the genes that we're looking for before deep cell sorting and after deep cell sorting. So our system was able to enrich these cancer cells based on just morphology. The last example I'll go through is using morphology as a monitor of cell health. So here we induced cell death in a variety of different methods with 2 different concentrations of staurosporine and by heating the cells. And as expected, the cells die. The top panel shows standard kind of cytoxin and Nexin monitoring of cell death. And you can see that based on the flow, the cells are dying. But when we look at the morphology, we can see that the cells are dying in much different ways. So we can basically follow the progression of the cell death through morphology in a much more granular and specific way than we can through just looking at cytoxin and Nexin. And that's reflected in the actual images of the cells themselves. You can see them going through the different states in cell death. So to summarize, the Deep Cell platform is a new system that is attempting to bring morphology into the same quantitative realm as other types of omics. It's an all-in-one system, and you can try it out with our applications lab. So we are pre-commercial, but we are accepting samples to our applications lab, where you can run a few samples and get a sense of the technology and potentially use that data to apply for other grants and things. And we are at booth 203 if you would like more information on that. We also are running a science challenge where you can enter to win a chance to do a proof of concept with us and generate a little bit more data than you would through our apps lab. And we are accepting submissions now, and we'll be citing later this month. So thank you for your time, and we're at booth 203 if you have any questions. Thank you.
Good.
Oh, there you go. Go ahead. I can just
Yeah.
give a quick introduction. So, good morning, everyone. Thank you all for attending. So, I have to tell you that, as talking to Rand, that there'll be more questions here in our presentation than answers. And the goal was to pick brains, and we got a lot of brains here. So, thank you. So, I want to thank Julie OJ for working with me when this presentation wouldn't have happened if Julie didn't help. So how we plan to do this is to kind of go through our current status of our cores, talk about some of the challenges that we are facing, and look at, on a structural basis, are there some alternatives? Then we will look into what the current models are out there. We haven't done an extensive search, but we have gathered some info, and then wrap it up with a discussion on what does it take to face these challenges in terms of structure. So, thank you.
Good morning, everybody. It's great to see everyone here. Thanks for showing up. And I want to thank Raj for inviting me to participate in this session. It's actually an interesting topic, how cores evolve and how they continue to evolve. I can tell you one of the first things that, I shouldn't say one of the first things, but one of the earliest things I learned in my career was to always be looking over my shoulder to make sure that I remained relevant. I had a really good mentor many years ago at University of Illinois in Urbana-Champaign who used to say to me on a frequent basis, Julie, you're doing great. We love what your core is doing. We love what's happening, in the flow cytometry world, but we may not like you tomorrow. So that to me said, wow, I better make sure I'm always on my game and that I'm always evolving with what the research needs are. So what we want to talk a little bit about is kind of how cores evolve, how they need to continue to evolve, where we think we are today, and where we think they're going to go. I found this diagram and actually a definition of the sharing economy, which is something that they said was actually relatively new, this term, the sharing economy. And what they mean by the sharing economy, at least according to the Oxford Dictionary, is it's an economic system in which assets or services are shared between private individuals, either free or for a fee, and today it means typically by means of the internet. Okay, so you can see how modern this definition is, actually. And there's a couple of indicators up here that I got from Wikipedia that says actually what the sharing economy is. And the first one it says was somebody first mentioned it in 1978. So I don't know how many of you in the room, I suspect not as many as I would think, have been around as long as I have in core facilities, but we certainly know that technologies in science have been shared well before 1978. I think some of the first flow cytometry cores actually were established prior to that. So, but then there's this interesting statement that says that the term sharing economy began in 2007, right? So I just wanted to put this up here because we actually have been in the scientific world using and participating in the sharing economy well before 2007. I think today this means things like car sharing, ride sharing, that sort of thing. But scientifically, you know, we've been doing this for a long time. The positives and the pros that we have found around the sharing economy and what's actually defined in Wikipedia is reduction in unused value, right? So we think of that as excess capacity that we have with technologies. We know that if we put technologies into a space where they can be shared in laboratories as opposed to in individual labs, the excess capacity that's available can be shared amongst lots of researchers. So this is not new to us, but it's actually a real pro, a positive for having things in a shared space. Access to goods without the requirement to purchase. That's definitely where our researchers benefit from putting technologies into a shared space. They don't have to purchase their own. An increase in quality of products and services, and strengthening communities. And I think that that's actually a really key point that we have found over the years in core facilities, scientific core facilities, is that creation of the community, the ability to actually not only share technologies, but to share thoughts and ideas. I used to, and still refer to core facilities as the water coolers of the scientific labs, right? It's where graduate students, postdocs, sometimes PIs, come together in a space and say, oh, what are you doing with the technology? What are you doing with that instrument? How are you doing that? And how are you using that today? And so building that sense of collaboration and informal conversations in cores is something that I think has been really, really important to the research community. In science, specifically, deep knowledge experts on the applications as well as the technologies is definitely a positive, right? This is a real pro of having core facilities. And I don't need to convince anybody in this room of that, but I think that that is having defined experts on technologies that can actually share not only applications and how to use that technology, but how to develop that technology further has been really positive. And so having very specific technology-focused core facilities, I think, has been really important to the community. And of course, the other positive is the elimination of the black box approach, right, to, or the kit approach to scientific assays. Today, I think there are a lot of there's a lot of kits out there. There's a lot of graduate students, a lot of postdocs who don't necessarily understand what it is that's going into the kits that they're using, the reagents that they're using, and then the instrumentation that they're using. And if they don't, I shouldn't say that they don't understand it, because I think they work they work closely with core facilities. But if they don't, I think people feel like they just take an assay, follow the directions on the box, put it into an instrument, and get some data out. And if you don't understand what's happening in the instrumentation, you can't fully understand the data that you're getting. And so I think that's where having core facilities with dedicated experts has been very beneficial to the research community as well, is we understand what that technology does, we understand how the different detection methods and analytical methods are generating the data, and then we understand how that data is actually interpreted and can help manage that. And of course, we all know one of the biggest positives of having instrumentation under the direction of individual experts is that you have better maintained instrumentation. You don't have to walk in and find, the piece of equipment no longer working, nobody told you, my experiment is ready to go, and somebody broke the microscope and didn't tell me. So I think instrumentation maintenance is really a positive. We also see core facilities have developed over the years in many different areas of science. It's not just the biological sciences that are represented here, but you can see on this screen, there's a number of different institutions, and I can't see without my glasses what it says here, but the different institutions, there's a website specifically for physics core facilities, so in the hard sciences, Department of Chemistry there at, I can't tell you what institution it is, but but hopefully you guys can see it. There's chemistry and biochemistry in the bottom corner, bottom left, and then of course I can see UC Irvine Biological Sciences in the corner. But even though the crowd here is mostly biological sciences, I think that's definitely changed too, and there have been actually core facilities in the physical sciences for many years as well. I see Carolyn nodding her head, my physics fans here. So I think that, cores are developing, right? Have been evolving over the years. I think what the development of specifically focused core facilities has been because we needed expert personnel with very specific customer service skills. Not every scientist has great customer service skills or great personnel interaction skills, and so core facilities, I think, provide this. They provide, but within the cores, we also need continuing education, right, for individuals to maintain their expertise in a particular area. And we also have to recognize that individuals that work in cores have to be disciplined generalists. They have to know the biological or chemical or physical systems that they're working with in general, but application specialists. How do we apply the technologies and the tools that we have and develop to a broad variety of different scientific structures and scientific projects. And so these are where I think, focusing core facilities and what we've done over the years is in developing, over the last 40 years, developing very focused core facilities has been really important. This is some of the challenges, though, that we face in developing in our core facilities today and that we've seen develop over the years, right? One, I think everybody can recognize that it's sometimes challenging that core scientists are seen as service providers only, as opposed to research professionals and research collaborators. And I think that this is something that is changing but needs to continue to change, right? We still need to see a little bit more of an evolution. There's definitely things that core facilities do, platform providers do, that are very service-oriented. But being part of a pipeline now of projects as they're developing, I think we see much more of a continuum in different core facilities or platforms being used in projects. I think we need to continue to evolve the language and the approaches that we have for core scientists to considered research participants. We also continue to see excess capacity in cores. I think that this is something that happens because we have seen sometimes very specific specialization within different core facilities. And depending on the size of the institute, sometimes that kind of specialization can lead to actually excess capacity as opposed to broad use of the technologies. I think the other challenge that we have in cores today is that we have instruments or investments, I should say, that are focused on the needs of the many versus the needs of the few. And I think that that's really important, and I think that kind of investment is necessary. But I think that what it does is sometimes limits what kind of investments are made in shared technologies that are really cutting edge or bleeding edge. And so I think this is an area that we have to focus on where cores can continue to evolve. And so what I'm going to do is turn the mic over to Raj, and he's going to give us a little bit more of a where can we go from here, and what are some possibilities for cores to continue to evolve. And then we'll come back and we'll have a great roundtable discussion.
Thank you, Julie. All right, so before getting, this is an example I wanted to show. So this is a series of thoughts, really, and my experience. So my background is I was leading the Shared Resources Program at Fred Hutch. Oh, it's a very robust program. Probably some of my previous colleagues are here, maybe not. Oh, there you go. It's an amazing program, very robust, very big program. And something that I noticed there was when we have multidisciplinary new technology, it was not easy, at least in the beginning, right? You see that chart, how many publications are coming in spatial transcriptomics 3-4 years ago, but it was starting, that's peak. It was not sure which, because we are all about specialization, expertise, and we have different vertical structures. And when you have a multidisciplinary challenge that's there, it's not very easy to slide into solving that problem with the structures that we have. The other thing that I have noticed is the peaks and valleys that Julie mentioned that we have. That's a challenge for us. We have one core. There is time when everything is crazy, and there is time when things are slow. But is there an opportunity when it is really hard for a core and a little bit extra time for another core to kind of help out? Or are we stuck in these specialized compartments? Are there opportunity for supporting a cross? And so with that, and then of course right now we can see one example I give is the Chang Zuckerberg imaging scientist position. If you look at the position description, it's very interesting. It's not a microscopy position. It could, you can have a engineering background, you can have a math background, you can have computational microscopy. So it kind of shows us how multidisciplinary things are changing at a higher specialist level as well as maybe a tech level. So I took this example to show how many cores really in our standard structure, how many cores can be part of this, right? You can see the pictures. I'm not an expert in spatial transcriptomics. I just picked a picture from a nice publication. My goal was to show that, there is histology, histopathology involved, there is gene expression genomics involved, there is computational biology involved. And if you really look at what a PI may have to go through in an institution, maybe 5 cores or more, right? If you need the histopathology. If you're trying to build a new antibody, you need the antibody development core, microscopy, depending on which vendor you're working with, which technology you're working with, genomics and computational biology. And if you look at the structures, how we are typically structured, we got these, I wouldn't call silos, vertical structures, right? Usually they don't cross-react a lot. There is collaboration, it's expected, but they are, in each vertical structures. And when I look at a PI, this is how the process goes, right? There's a lot of interaction. You get the antibody, you work with this core, work with that core, maybe talk to the director, talk to the person, and there is a lot going on there. And the question is this something that is desirable as we go more and more into multidisciplinary? There's a lot of talks going on, multi-omics, I think is going on right now. There is another talk, I think, later today about single-person cores. That's the other interesting topic, because where I'm coming from is I'm at Seattle Children's now. Part of my job is to build some cores. And I'm looking at efficiencies, right? Instead of building, do you want to build, and that's the origin of this talk, because do you want to build it like this, or do you want to build something different? Is there an opportunity for cross support across different cores where you can actually have get closer to optimal capacity in terms of people, not just equipment, people and equipment? So again, I'm not an expert in organizational structures, but did read a lot on this. And there are 6, 7 organizational structures that publications describe, experts in this area describe, but most of them fall between the hierarchical vertical structure and the horizontal structure. And then there's called the divisional structure. There are international type of, with big companies, they have different other structures. And everything in between, we're going to call matrix structures. Something in between. It could be slightly different for each situation. So the matrix model concept started in the 1960s in aerospace industry. And note this, because they were facing the challenge of multidisciplinary approach to build a plane. Because technology was progressing, there were all specialists, lots of specialist groups. They had to get these things together, build a plane, and they came up with this idea. And everybody jumped on it. It became a big deal. Everybody wanted to go matrix models, and there was a big backlash because a lot of places it failed. In 1970, there's a paper in HBR, if you want to look at it, focused purely on why this is a terrible model. And 50, 60 years later, I'll show you another slide later, that majority of US workforce, based on a survey, are in some level of matrix model. They're working in some level of matrix model. So it is there, it is popular. I can't speak how popular in our core facility world, but it's something I would say we should consider. I think that's my slide. I think we go, okay. So I want to just make this point before we shift to the, whatever models that we could point out for now. I mean, what the real world models that Judy's going to talk about. But these are the key aspects of an organization or health of an organization, right? On that side, we have the accountability, responsibility, decision making. There's somebody in charge, of course, efficiency. And on this side, collaboration, communication, innovation. I mean, these are all key things for core facilities. And in the end, job satisfaction. So how do you think about different structures and making sure that all these key points that's important for organizational health, we are keeping them going, not compromising any of them. So we would shift to some real world models.
Yeah, so I'm going to talk about a couple of models that we've identified out there. Roger and I were trying to think of organizations that we know that have moved away from the single core, single discipline focused model into something different. And for those of you that are in organizations that have already kind of shifted in that way, we really want to hear from you when we're done talking up here. The first model that I'm going to show you is, So how do I not make that? Sorry, I don't know how that happened like that for those, the animation on that, but yeah, it automatically went through.
Oh, it's going itself?
Yeah. All right. That's because I got these slides from my colleague, Max Krummel at UCSF. Max, I don't know how it's set up in there. And actually, I had them on a Mac, and now they're on a PC, so I'm not quite sure what happened there. But anyway, any of you from UCSF, anybody here from UCSF in the crowd? No? Oh yeah. Well, John, you're no longer from UCSF, but Rochelle, yeah. So Oh, yeah, and Val, great, back in the corner. UCSF a few years ago, on their Parnassus campus, in their immunology group, developed a structure that they called, it's called ImmunoX.
Presentation mode.
Sure. Go back to, yeah. Okay, so we're not going to do this in presentation mode so they don't move, okay? But you can see they developed institutionally on their Parnassus campus something that they called collabs. And they were designed to focus specifically on the immunology-based community on their Parnassus campus. And they were, you can see that there are 5 different organizational structures here that were designed to support what they needed. They are the Disease to Biology Collab, Flow Cytometry Collab, Biological Imaging Development, Genomics, and a Data Sciences Collab. The way that they envisioned that they would be used is they have an entire pipeline, and an investigator comes in and looks at, from a project basis, what it is that they need and how do they integrate these. Is that because I'm not in presentation mode? Okay. Let me just show this last slide and we'll go back into presentation mode. But what they actually showed is, or the way that the Collabs project works is that there are individual projects that come in that have different specialists that work on different parts of the project. And so even though they are within a particular technology discipline, for example, the flow cytometry collab, which is the second column here, there are cytometry specialists in that core that work across different projects, right? So one might be dedicated to a specific project. They attend project lab meetings, they participate in all of the conversations about the science in that entire pipeline. And so while they are core specialists, they also work across different projects. So that's the UCSF model. And we're going to go back into presentation mode so that we don't have to deal with. This is another model that I want to show you. And I'm not going to talk about the pros and the cons of these 2 models because they aren't at an institution that I participate in or a part of today, but I wanted to just show them as possible models. This is how the Stowers Institute restructured their core facilities a few years ago into what they call technology hubs, or as identified here, 7 scientific departments. So Stowers Institute has all of these different core facilities that existed prior as individual cores, and they have now restructured them under different technology hubs. So you can see the first one, model organism research, includes what was initially independently laboratory animal services, reptiles and aquatics, and transgenic and reproductive technologies. And all of those structures are now organized under Heather Marshall, who is the director of the model organism research organization. So again, anybody in here from Stowers? No? Okay, so I can talk freely about their model? No, I'm kidding. This, again, is another way to kind of define, we have a technology hub. We're thinking at the Salk Institute about how we may be restructuring some of our facilities as well, because, as Raj mentioned, there's all these interdisciplinary technologies that really cross boundaries now. And it's hard to have, to necessarily go to just one facility or one core director or one core specialist when the technologies span multiple different types of, or the projects span multiple types of technologies. So what we want to think about is other ways that we can develop cores into more structural hubs, into a community, one hub maybe. I don't know what the model should be in the end, but that's what, excuse me, what I think we want to have that conversation is whether or not anybody else has developed a matrix model, thinking about matrix models, and then have that conversation across the group.
So I keep calling this matrix systems. I mean, not in a pure structural way, but something in between the vertical. Something different where people between core facilities interact more closely. So the UCSF system, looking from outside, we did try to get representatives from UCSF. They were ready to do online, web-based. We didn't have an opportunity to have a web-based presentation, so they couldn't make it. But looking out from outside, it looked like the classic matrix system. And I did some search online. But I have nothing to do with this conflict of interest here statement. Nothing to do with McKinsey. But they did a survey of our 2,500 employees in the U.S. It's a representative survey. And what they found, and it was about the matrix system, how is that working? And what they found is that the employee engagement, the level of they have 4 classifications here: the normal, non-matrixed, and then slightly matrixed, matrix, and super matrix. I think that's a good way to kind of say, put that. Super matrix means you got 2 bosses, you report on a daily basis, and then it comes down from the dotted lines in some places. But anyway, what they found is that the employee engagement, which is an important aspect of job satisfaction, it actually goes up. And I can tell you, so I come from Seattle Children's, and I moved from Fred Hutch, which is not a matrix organization, and I come in the new place, and I look around. We have our faculty, Seattle Children faculty are all UW faculty. The UW HR, University of Washington HR deals with them. But everybody under them in each lab are Seattle Children employees. Seattle Children HR deals with them. Oh, and by the way, Seattle Children's Research Institute and the hospital IT and many other functions are actually coming from the hospital, and they have dotted lines too. So we live in a fully matrix system in Seattle Children's Research Institute. And what I observe is that, this very young organization, 15 years, and now I think we are among the top 4 NIH-funded pediatric research organizations. So not bad for 15 years. And if you ask the employees, you will hear a lot of complaints about the challenges of the matrix system. But something is working. Don't want to go into details because I don't know how to analyze that. So coming back to this. So what they found is that in a matrix system, based on the survey, if you look at, and that's a McKinsey proprietary org health index concept. And the most of those topics that you see, the pillars that you see are very familiar with us when you look at about organization. And they found that all these are doing well in a matrix system, but some challenges that you see with the matrix system, which is why it initially came up and then was criticized, is mainly because of these 3 key aspects: the leadership, control, or clarity of roles and responsibilities, which are critical for any organization. So how do you make matrix organizations work? I'm jumping into this to, if you are thinking about something different other than the siloed structures that we have or individual vertical structures that we have, the biggest, there are 4 pillars, the where, what, who, and how. The one is clarify the reporting relationships. In fact, when we talk about matrix systems, we have these dotted lines. What they recommend is take that out, right? Make it very clear. Dotted means it's not clear. And that's where every problem starts. So have very clear reporting relationships. It might be multiple bosses, multiple roles and responsibilities, but make it clear up front. The roles and incentives, make it clear. Again, more clarity. Be very proactive about bringing clarity. And also remember that the balance of power keeps shifting in a matrix system. It's not that settled system. and it's very dynamic. So people need to learn, and these are the fundamental education that's needed for everybody to thrive in a metric system. And have clarity in processes, and there's some examples here, right? You can have some operating principles. And usually what happens in a metric system is it allows the employees at the front, to make decisions, which is very enabling. Not the boss doesn't have to make decisions on everything. So make that very clear, and that's how prioritization happens and escalations. So you have to have some clear operating principles that guide people in that confusion of metrics, so that there's easier solutions, and everything doesn't go to the leadership. And so it will be, if it is not implemented right, the leadership will be struggling with, and that's one of the biggest, if you read the 1970 paper, that's the biggest concern. The leadership ended with conflicts to solve, like 300%, right? Everybody's escalating everything. And now, how do I do this? Now, who do I report to in this? And so all that need to be clarified up front as much as possible. And the most important part is the leadership mindset and skills. And I think that's, if you're thinking about a different structure, I think that's where us as core facility, more sciencey people need a lot more learning, because it is the leadership skills. And this HBR article, they did a survey, and what they found is that the critical leadership skills needed for success in a metric system are the most rare ones. It's not that common. So the empathy and influence, right? It's not your power. It's not like how, everything goes as I say. You have to use your influence, not the direct power. Conflict management and self-awareness. And those areas are important to be successful. And so in summary, if you want to change the structure to adapt to the challenges that comes in an organization that is vertically structured, it needs a very strong groundwork and preparation, a very strong leadership commitment, not just the leadership of the cores, but the institutional commitment, organizational commitment. It needs a lot of support and training, because the transition, as shown in the history, is not easy. You can't just jump in and say, from tomorrow onwards, we're going to be a matrix organization, multidisciplinary. It's not going to work. And the last is, it's not a structure, it's a frame of mind. So it is very dynamic. It's day to day, things change. And that's where the ability of the employees, the leadership to kind of, roll with the punches is very important. So with that, really, as I mentioned before, it's really having a round table and getting input. We would love to hear from you if you are aware in your institution or anybody have a different structure, experimented with different structures, because I believe that this is a longer conversation that probably we can start now. So we want to thank the UCSF Collab leaders, as well as Stowers, Heather from Stowers, for sharing those slides. So with that, I want to open it up for discussion.
So I don't know if, these microphones do work. Okay. So this is the audience participation time. Anybody here work in a structure that has developed into a more multidisciplinary core? Got a couple of hands. Interested in sharing your experience?
Hi, everybody. My name is Caroline Denef, and I'm a director of a core facility at Colorado State University. Thank you, Julie. And I forgot your name, sorry.
Raj.
Raj, for your wonderful presentation, because this is very dear to my heart. I was a core director of kind of a chemistry-based core facility at CSU. And in 2020, we merged with a number of cores that were from very different disciplines. So both in life sciences as well as materials characterization, synthetic chemistry, and we brought these cores all together. They belong to different departments and colleges and university, and The idea was, let's bring these cores together. They serve a similar mission. They're operated very similarly, but they're kind of reaching, they're going beyond the boundaries of the departments and being used by such a wide, broad user base on campus that the way they're funded doesn't make sense anymore. We should bring them up to a more central level. So we were brought under the vice president of research. So now I have a core. I became the director that has about 5 or 6, I would call more mini or sub-cores that are all led by specialists in their area, and they're called lab managers now. But we really see the benefits of this, and I would say in multiple ways. I would say, first of all, my staff, it's a group of 15 scientists who are all experts in various areas, in multidisciplines. They all work together, and they approach projects way more collaboratively. In the past, they would hold on to projects maybe because they want to own it and take the money. They need to recover their costs, but sometimes the project would belong better in another core. We have lots of redundancies in mass spectrometry that we merged and kind of rethought other efficiencies that we can create and make sure that we approach a project much more from the customer's perspective instead of, I just want to do it because we need the money. So financially, we're one structure right now with one recharge account. I would also say that because we kind of created this multi-discipline structure, that we also were able to streamline a lot of our processes. The way we bill, the way we quote and communicate with customers is much more handled from a project management type of approach. We work in teams collaboratively where an inquiry comes in, one person leads it and brings all the experts within a team chat, and we can talk behind the scenes before we really reach out to the customer, because in the past we would see that customer being just thrown around to different cores, and now it's much more done collaboratively. I don't know if it's really because of this merger, but we've had some successes with funding. With NSF MRI, we got 2 big shared instrumentation proposals funded, and the reviewers really commented on the way the core was managed and showed some interest and curiosity in that being a demonstration of, let's say, getting really good institutional support for bringing these cores under a more central model. Challenges, I would say, it is hard to manage a lot bigger group. So I know here there's a lot of solo staffed cores who have challenges with being one in a core, but managing a really big core with lots of people is also really hard. There's issues around equity. We had to bring cores in together that came from different units, and everybody was paid very differently, and so having to resolve equity issues is really hard. And that took a really long time, but I feel now that we resolved those issues, people work more, they're less competitive, or they tend to just be more of a community, I would say. So I like your comment about that. So yeah, I'm happy to talk more about my experiences, but it wasn't easy to bring different units together, but in the end, I think it really worked for the best. And as a core director, I'm much more of a generalist now. I know a lot about the different technologies, but for me, it's easier to speak to customers who have needs that are cross-disciplinary, because I can quickly connect them with the right people. So I hope that helped.
Yeah, that's a great real-world model from somebody who has developed it. So thank you so much for sharing that, Carolyn. And if anybody has specific questions about how she did it.
I actually have a question for you. So one of the challenges that when I think about it is, in our core structure, typical classical structure, these positions are part of career growth, right? And when you make it more flat, relatively, how do you address that? How do you address, I mean, that's an HR, right? You have to work with HR, but how do you, so the concept of career development and career growth has to be, it has to change a little bit.
Yeah, that's a really good question, and it's something I'm still trying to figure out. I would say one of the challenges we had was that every one of the subcores each had a director, and so bringing them together, the idea was a little bit, we don't need 5 directors. Let's create efficiencies there, and it's really hard because you really can't demote people. It's just a tough thing to do. So we created more associate director roles, or we're working on that, and give people titles that are more reflective of what they're doing without having a specific director title. I think in our structure now we have more managers, and they're very happy to give the administrative duties that they were taking on in their cores away and let the director do that. So I think they're actually feeling the benefit of having more time to really be a really good operational manager and have more time to develop professionally in their area of expertise. I do get questions every year about when can I get promoted, or what does that look like. When I hire new people, they always want to know what their career path is. But I don't have an answer for that. I would say I would love to see at CSU a more defined job classification and job families that that really speak to the titles of the core people. Right now, all my staff falls into research scientist and research administrator, research assistant job descriptions, and they're not very reflective of what they're actually doing, and so the next level up is really hard for them to reach because it's not really in the description of the job. So I don't think we're there yet at my university, but we try to just base it more on experience, seniority, where you are in your career, and just make the argument to the leadership that a person is really ready for promotion, and I fight. Yeah.
I think
Thank you.
there is a session on core structures, core personnel yet within today or tomorrow. So that's actually a good segue into that. Can we hear from the other individual who also is working in kind of a matrix model or a shared, an evolved core, and then we can see if we can compare and contrast some
So my name
ideas.
is John Wolderman, and I work in the University Instrumentation Center at UNH. We have been a university-wide core facility since 1973, and we were put under the research office when it was conceptualized in 1978, and we've been there ever since. We are multidisciplinary. We have EM, we have NMR, we have flow cytometry, confocal microscopy, X-ray microscopy. We deal with material science, mechanical engineering, biological, chemistry, the whole 9 yards. I echo a lot of what you said. As far as personnel, we're small. There's only 7 of us. We're scientists and engineers. We have 1 director, so that makes it easy. There's not a lot of room for growth per se. We do have a track system for analytical instrumentation scientists and engineers, so there is some movement. But it's been good because we're non-political, if you will. We don't fall under a different college or within a department. We work with many different departments and colleges across campus. We also have services that we offer as far as repair services and calibration services as well from the engineering point of view. We deal with internal customers as well as external customers. So we have a lot of industry that we work with throughout the year. And that gets back to some of the funding models and helps us to maintain the equipment with that type of income. And we're being centralized has been good. When I did, back in 2006, I looked out across over 300 different land-grant universities across the U.S. for a similar core facility like we had. And at that time, I found only 3. Mm-hmm. Since then, that has grown, and the granting agencies have been pushing that as well, looking for more cores that are centralized through funding from the universities, because they get more bang for their buck, if you will, for the granting of instrument money or whatever that they give. A lot of our instruments, we have about $6.5 million worth of equipment right now that we're responsible for, and almost all of that has either been funded by NIH or NSF.
Great, thank you. Yeah.
Since I couldn't see the faces over there, I'm going to stand up. Hi, I'm Jessica Back. I am at Wayne State University in Detroit. And I wouldn't say we're a functional, centralized facility. So my core is one of the siloed cores at our university, but it is a little more multidisciplinary than a lot of the others. So we do microscopy that traditionally had been light microscopy, but we just inherited an electron microscope. We do flow cytometry, which is my kind of baby. And then we do small animal imaging, which includes PET, SPECT, CT, optical, and now optoacoustic. And I say and now because when they're recruiting people and they need a new instrument, it seems like we get them all. Oh, and we have an irradiator. So we have 25 instruments across 5 different buildings, and there are 7 of us total. And the university so we were combined, the microscopy imaging and the flow cytometry were combined in 2008 with AARA funds. So we have 7 staff total. That includes my director, I'm the associate director. And then we kind of segregate our staff. So we have 2 animal imaging specialists, 2 microscopy specialists, and the 2 flow cytometry specialists include me. So we as a core, I think, operate really well, but we also have challenges in that kind of everything comes to us, which I know sounds really crazy, like, oh, you're getting new instrumentation. It's great, except I don't know anything about optoacoustic imaging, and now I have to learn it. And I didn't have any say in that capital equipment investment other than we have this recruit and we don't put instrumentation that they need into their own labs. It has to go into a core, and it's going to go into your core because that's where it makes sense. And then we have to kind of make it work. And so my imaging staff are predominantly trained in PET and CT. And now we're throwing them into technologies that they know nothing about. And so that is a real challenge for us when it comes to those new things. So my boss will be retiring in a couple of years, and I'll get to take over this monstrosity. And I'm already thinking about where we can have efficiencies. There are several scopes. I love him, and he likes to hold on to things for a long time. So we have, those of you who are microscopists, I still have a Zeiss 410 in my lab. It's finally been decommissioned, finally. But we had a faculty candidate on Friday, and he showed it to him. I'm like, no, it's decommissioned. So, I'm already thinking about where we can make some efficiencies, because as you can imagine, with all that instrumentation, even though we're an R1 university research institution, we are never anywhere near our capacity on anything. And so that's a problem for me, because then how do we show that we're useful if we are not providing a useful amount of instrumentation time? We also have another large core like that on campus. It's our chemistry core, and they have all the mass specs and NMRs and those kind of things. And then we also have, because they have mass specs in the Lumigen core, we also have a proteomics core with mass specs and a lipidomics core with mass specs, which I look at and I think, yes, they're very different usage of those instruments, but also why do we have all these mass specs in all of these different buildings across campus? So we all now fall under the Office of the VP for Research, but I also serve 2 masters because we have a cancer center. So we're a cancer center support grant supported core. Not all of them are. And so it's, like I said, I don't think we do it well. We're trying, we're getting better. And we have a new VP that I think is starting to think a little more strategically about these things. I've also lived through that short-lived period of time when the Cancer Center support grant required you to be clustered, the cores to be clustered. So any of you guys who went through that understand what I'm talking about. And we were clustered with proteomics and animal resource or animal modeling. So yeah, I'm curious. I'm really excited to hear there are others like us, because I sometimes go to flow meetings and I feel completely outside of the realm. But I would love to get ideas on how to do it better.
Yeah, I think Jessica brings up a number of points, and I think the first one that's really critical that I struggle with when I think about this changing model or the evolving model is the personnel expertise, right? How can you I mean, it's great to, like I said, always making sure you're relevant, right? Making sure that you're evolving your skill set so that when the technology that you're an expert on today becomes obsolete, you don't become obsolete with it, right? That was always my fear, right? I wanted to make sure I always had a job. But how do we actually take the time or find the opportunities to learn new technologies while we're busy managing or operating the technologies that we're responsible for today, right? And so I think part of this has to be an institutional leadership approach. So those of you that are in more management and administrative positions, making sure that there's time for individuals, an opportunity for individuals to get some additional training, to see what's out there, to learn what new cutting edge technologies might be coming out there, I think is really a critical thing. And so if you're not in that position, appealing to those that are in those positions to make sure that you have the time to learn new skill sets and new technologies is really important. Anybody have any thoughts about how they've done that at their organization, or show of hands on how many people struggle with trying to find the time to learn new technologies? Anybody besides Jessica and her team? Yeah, I see a lot of people who, yeah, absolutely. Yeah, comment back there? Or just raising your hand to say, yeah, I'm one of those.
I'll comment and say, just to add to it, that there needs to be funding for it, because one of the challenges, I'm at a wealthy institution, so in that way I'm very lucky, but one of the things they don't spend on is sending us to any kind of courses to learn things, or sending us to meetings where we can learn things. And I think so that's very important for anybody here who has control of a budget in that way, to realize your staff, because I'm a staff, not a director, need to actually have dedicated time to learn things, and that includes sometimes maybe $3,000 to go to a class.
Yeah, great point, really great point. Any other thoughts about how to do that? That type of training, of course, can be included in recharge rate development for those of you who are, have to think about where do we get the funding for that. Joe?
Hi, Joe Dragavon, CU Boulder. I just wanted to back up for a second. We're very siloed for all our cores, and we have no centralized support, but the university is exploring it. How did people transition from no centralized support to some sort of centralized structure? Because I see the conversations like, oh, you're centralized, you can do all this. You're uncentralized, you can not do all this. So how do we transition there?
So,
and I'm looking for any thoughts.
Joe,
about
can you clarify, for everyone to be on the same page, when you say non-centralized, you mean in a departmental core?
Yeah, all
right?
the cores rest at the departmental level.
Within the department.
Yeah, so that's actually a very different conversation, and lots of sessions have been held in the past about how that's happened. I can tell you, when I got into the field of flow cytometry a long, long time ago, there were very few centralized administrative structures. And when we developed that at the University of Chicago in 2000, I think I could count maybe across the country 5 to 10 institutions that had done so. That has happened. That evolution has happened, and is something that continues. But there's still organizations out there, obviously, like yours, Joe, who haven't made that leap. Some of that has actually come from the ground up, right? Where cores kind of organize themselves. So if you're not in an institute where you have an administrative structure, I have found that self-organizing is very beneficial. And that actually allows leadership then to see the benefits of having common conversations across multiple disciplines. And I think that is what then leads us to having, these matrix organizations under, just one scientific structure as well. Now, whether we continue, whether that's the best course of evolution for the cores into, scientifically matrixed organizations, I think is yet to be determined. But that would be my advice there.
I can briefly share my experience, Joe, because, I moved to Seattle Children's, and one of my goal was to centralize cores, and part of it is to get the faculty with you. And so the first 2 years, honestly, was to talk to faculty and finally do a survey where everybody's screaming for centralized cores, and also clarifying to the leadership that if you want to recruit top talent, it's not just about the money. It's about the reputation. It's about, like yesterday's presentation, it's a strategic investment. And you need to have that both sides. The faculty, you will see some senior faculty who's been there, being successful in their own ways. But you got a lot of junior faculty coming. That's where I find all my strength. And they want this. So you had to work with the faculty on one side. On the other side, you had to convince the leadership that this is the way to go, or we're going to be behind.
Just to address Joe's question, the two institutions where I've done this for a career, it's been the carrot model that brings them to the table. You want, if you want subsidy, institutional subsidy, then you need to, join in, into a centralized model. You want an iLab seat? Well, then we're also going to handle your administration. Access to CapEx funds or OpEx funds. That's always what has brought cores, from the departments to a centralized oversight for us, and at my current institution and the last one. And right now I have an analyst and a billing specialist that work for me. And that's something that is tantalizing to a core director, is someone who can handle my billing, worry if the grants are expired or not, and all I need to do is push done. That's what brought them to the table for us. Sorry, I'm Matt DeVries, City of Hope National Research Hospital in Duarte, California.
Thanks, Matt. I think that there's a, I think that as Matt was talking, I was thinking about this organizational change, right? The type of organizational change that when we are in siloed cores and specific discipline cores, right? We're thinking about that potential evolution into a more matrixed organization. It's the same psychological thing that happens, that speaks to Joe's question, right? About how do you go from, departmental cores into an institutional core. A lot of it is about wanting to hang on to power and control, right? And that's a tough thing for all of us, right? We don't want to be absorbed into a matrix core, because what does that mean for me? Right? What does that mean for me and my team, right? What will I potentially not control anymore? I think the same thing, when we think about departmental cores moving into a central administrative core in an office of research or in a VP for research structure. What is the departmental chair giving up? What is the dean giving up when they're moving cores from their school or their college into, the bigger administrative structure managed by the provost or the chancellor? It all comes down to control and power, right? I think those of us that work in cores and have been in cores for our career recognize the importance of sharing. Our job is to actually continue to communicate how we can actually benefit from those shared infrastructures as opposed to siloed infrastructures, right? And recognize what are the benefits that we gain, and how are they better than what little control we might be giving up, right? And kind of get rid of that fear. But it's a very tough psychological thing, right? We all have to deal with. So one of the things about this organization, ABRF, people in this room, is we're a great therapy group, right? So lots of colleagues out there who are going through the same thing, but I think we as a community actually can dictate that. So 2 comments.
Hi, Julie. Thanks.
Hey, Jill.
This is a really great session. So I'm Julie Rothicker from Melbourne, Australia, and I've been coming to ABRF for a number of years, and I think it's sort of really helped how we've evolved in terms of our core facilities, and absolutely at the same sort of stages. I think this particular session was interesting because we're now going from an evolution of having a central office and having one for a long time, and we will keep that, and there's a lot of benefits to that. But putting in really strong policy and governance so that we actually don't have to bring our cores up to a central level. So we're actually now moving them back into where the users are, because we're finding the narrative becomes us and them.
Yeah.
Department central, and losing that buy-in. So then the challenge is, how do you bring together multiple cores across departments but still have an ownership within a department? Where should it sit? But how do they still form the structure within the central sort of VP office? So that's something we're doing at the moment. It's sort of interesting to just see how it's evolved, but I'm not sure whether it's the right thing or not, but that's sort of, I think where, you know, if you've got good oversight, you've got good buy-in across the university and governance, and I resonated with get rid of those dotted lines and be really clear on responsibility. I think it can work. So do you have to bring everything up just to make it easier is probably my question.
Great. Well, we'll get an update from you next year. Bill, do you want to give one last closing comment, and then?
So that means keep it short. Okay. Bill Hendrickson, University of Illinois, formerly. When I came into our core organization, it was the opposite. We had a core organization that had started in 1948 with Air Force contracts for space research. And the problem there was it had become siloed itself. So it was a matrix organization that had dissociated from the rest of the world.
Yeah.
And we had exactly that. The departments weren't buying in, and so cores were beginning to spring up. And so my job was to rematrix the core organization. And the one thing that can counter power, and that was what I beat my head all the time, it was power. It was, I own this core, a
Yeah.
department head didn't care. They were losing money and losing customers. And the one thing that can counter that basically is money. And so, you know, you go in and show them, you can stop losing all this money. I can pay this, I will hire your person and pay them 25% more than you're paying them. And I could do that because I could show a model where we would double our throughput. And so I think there are ways around that siloing, but I felt it was the balance between the department and the central. So I was paying the person centrally. The core might actually be located in the department. You hired a person that came out of that department. Then you had the opposite problem was you got to make sure that they're equally accessible to all departments. And so that was sort of my job, was to make sure that everything was equal and all of that. But, so you can have matrix organizations that sort of fold in on themselves. So there's this constant need to go out and make sure your connections, your dendrites and your everything is going out to everybody. So that's all.
Thank you. Do we have times?
Everybody still okay with hanging for a minute? I don't know when we're supposed to be done, actually, but.
Hello.
fun.
I think the point that I'm going to make is important because I'm not a core director, so I'll bring a different perspective. I've been working in my institution for about 18 years, and there have been all this restructuring back and forth without anybody asking me a question of what do I think about it, how is that impacting from my professional career to my job satisfaction, right? And what I see constantly happening is the directors getting more responsibility in things that they don't know much about because they have, they keep becoming, I'm sorry, I get distracted. I have ADHD, that's why. So what I see is that people making the decisions are not closer to the facts of what's happening. So we have been tossed in different spaces because they restructure and they decide that these two cores have to be next to each other. And then when it doesn't work out, then we move to a different space. I have had to cross-train people or work with people that don't know anything of what I'm doing. I have been asked to train in technologies and instruments that I had never had before. So just as most of you are core managers, think about your staff as your most precious resource and instrument, and ask questions and go from there.
Thank you. Thank you. I think we are at time, but hopefully, if anybody has more thoughts on this, I think this is a beginning of a conversation. That's how I see it. Hopefully, we can have more sessions in future and continue this. It's a tough one.
Right.
But
And thank
very
you for those
interesting.
last comments.